ಬೇಡ ಕೊಡಿ.
ಸೊ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಸೊ ಗುಡ್ ಈವ್ನಿಂಗ್ ಒನ್ ಆಲ್. ಸೊ ಇಂದಿನ ವಿದ್ಯಾರ್ಥಿ ಪ್ರತಿಭಾ ಶೋಧ ಯೋಜನೆಯ 70ನೇ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಶ್ರೀಮತಿ ಸುಮಾ ಬಿವಿ ರವರನ್ನು ಈ ಒಂದು ವೇದಿಕೆಗೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ಐ ವೆಲ್ಕಮ್ ಟು ಯು ಮ್ಯಾಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಸರ್.
ಸೊ ಅದೇ ರೀತಿ ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಾಗಿರ್ತಕ್ಕಂಥ ನನ್ನೆಲ್ಲ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರು, ಅಧ್ಯಾಪಕರು, ಶಿಕ್ಷಕರು ಹಾಗೂ ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳನ್ನು ಕೂಡ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಹಾಗೂ ಈ ಒಂದು ವಿದ್ಯಾರ್ಥಿ ಪ್ರತಿಭಾ ಶೋಧ ಯೋಜನೆಯನ್ನು ಪ್ರತಿದಿನ ಸಲಹೆ ಸೂಚನೆಗಳನ್ನು ಕೊಡುತ್ತಾ ನಡೆಸಿಕೊಂಡು ಬರುತ್ತಿರುವ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ ಕೂಡ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ವೆಲ್ಕಮ್ ಟು ಯು ಸರ್. ಸೊ ಈಗಾಗಲೇ ಸುಮಾ ಮೇಡಮ್ ರವರು ಹಲವಾರು ಉಪನ್ಯಾಸಗಳನ್ನು ನಮಗೆಲ್ಲ ನೀಡಿದ್ದು, ಅವರ ಬಗ್ಗೆ ತಮ್ಮ ಕಿರುಪರಿಚಯಗಳನ್ನು ಮಾಡಿಕೊಟ್ಟಿರೋದ್ರಿಂದ ಹಾಗೂ ಕಾಲಾವಕಾಶವು ಕಡಿಮೆ ಇರೋದ್ರಿಂದ ಅವರನ್ನು ನೇರವಾಗಿ ಈ ಒಂದು ವೇದಿಕೆಗೆ ಮತ್ತೊಮ್ಮೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್ ಸರ್. ಥ್ಯಾಂಕ್ ಯು. ಶುರು ಮಾಡ್ಬೋದಾ ಸರ್?
ಎಸ್, ಓಕೆ ಮೇಡಮ್, ಓವರ್ ಟು ಯು.
ಎಸ್, ಥ್ಯಾಂಕ್ ಯು ಸೋ ಮಚ್. Good evening, everybody. Today we are going to discuss about very important and very interesting molecule which is present. Before going to that, ನೀವೆಲ್ಲ ಕೇಳಿರ್ತೀರಾ ಜನರಲಿ a commoner ಮಾತಾಡ್ಬೇಕಾದ್ರೆ ಯಾರನ್ನಾದ್ರೂ ನೋಡಿದ ತಕ್ಷಣ ಹೇಳ್ತಾರೆ ನಿಮ್ ಕಣ್ಣು ನಿಮ್ ತಾತನ್ ತರ ಇದೆ, ಇಲ್ಲ ನೀವು ನೋಡಕ್ಕೆ ಸ್ವಲ್ಪ ನಿಮ್ಮ ಅಜ್ಜಿ ತರ ಇದ್ದೀರಾ. ಈ ತರ ಎಲ್ಲ ಹೇಳಿದಾಗ ಕಾಮನರ್ ಏನಂತ ಹೇಳ್ತಾನೆ? ಯೂಶಲಿ ಅವನ್ ಹೇಳೋದು ಒಂದೇನೆ ಮಾತು, ಜೀನ್ಸ್ ಅಪ್ಪ ಇದಕ್ಕೆಲ್ಲ ಅಂತ. ಅದು ಎಲ್ಲರಿಗೂ ಗೊತ್ತಿರೋಂಥ ವಿಚಾರ. ಜೀನ್ಸ್ ಅಂದ್ಬಿಡ್ತಾರೆ, ಜಸ್ಟ್ ಜೀನ್ಸ್. ಅಂದ್ರೆ ಜೀನ್ಸ್ ಅಂದ್ರೆ ಏನು? ನಾವು ಹಾಕೊಳ್ಳೋ ಬಟ್ಟೆನ? ಇಲ್ವಲ್ಲ. ಓಕೆ, ಅಂದ್ರೆ ಜೀನ್ಸ್ ಅಂತ ನಾವು ಏನನ್ನ ಹೇಳ್ತೀವೋ ಅದು ಆಕ್ಚುಲಿ ಹೇಗಿದೆ, ಓಕೆ, ಅದ್ರ ಬಗ್ಗೆ ನಿಮಗೆ ಕೊಟ್ಟಿರೋ ಅಂತ ಡೀಟೇಲ್ಸ್ ಇರೋದು ಒಂದೇ ಒಂದು ಪಾಠದಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾರೆ, ಒಂದು ಚಾಪ್ಟರ್ ಅಲ್ಲಿ. ದಟ್ ಈಸ್ ನೋನ್ ಆಸ್ ಮಾಲಿಕ್ಯುಲರ್ ಬೇಸಿಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂತ. Why did they call it as the inheritance here? Inheritance means a molecule which is passed from parent to progeny. Okay, parent ಇಂದ next generation ಗೆ ಪಾಸ್ ಆಗಿರೋಂತಹ ಒಂದು molecule. Okay, that molecule we are going to study today in a small topic, that is molecular basis of inheritance. So today we are going to learn about that molecule, which is that molecule which is part of, okay, which is part of inheritance. Okay, molecular basis of inheritance. Okay, basis of inheritance. Okay, now let us take up first. In order to understand the molecule, first you should know how it is, the structure of the molecule. Then you can understand how it functions. So first we will take up the structure of molecule. Okay, now when you take up the very interesting molecule which is transferred and carrying the genes, like carrying the character from parent to progeny, it is in general known as nucleic acids. They are known as nucleic acids. These molecules are basically nucleic acids. When you take up nucleic acid in nature, okay, in living system, if you take the whole living system, there is only two important genetic nucleic acid present. I use the word genetic here because it is transferred from parent to progeny. Okay, which is the two nucleic acid present? One is DNA, another is RNA. Okay, RNA we will take up in detail in some other classes, but remember RNA is the genetic material in viruses. If you take up the 5 kingdoms, then you will not see RNA as genetic material. You will see only one molecule which is as genetic material present, which is DNA. ಬಟ್ ನಮ್ಗೆ ಹೆಂಗ್ ಗೊತ್ತಾಯ್ತು ಇದು DNA is the genetic material? ಇದು ಎಲ್ಲಿ ಇರುತ್ತೆ, ಇದು ಹೇಗೆ ಇದು ಟ್ರಾನ್ಸ್ಫರ್ ಆಗತ್ತೆ? If you want to know about all these things, first you should know how it is. Okay, then you can come to know how it is transferred from one to the other. Okay, so today now let us take up the structure of DNA. Okay, what is the expansion of DNA? It is deoxyribo, okay, nucleic acid, deoxyribonucleic acid. Okay, how do we know that it is acid in nature? Many scientists have worked on this molecule. The first one to name is Frederick Misha. ಓಕೆ, ನೀವು ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕಿದು, ಯಾಕಂದ್ರೆ MCQ ನಲ್ಲಿ ಕೇಳ್ಬೋದು. ಫ್ರೆಡ್ರಿಕ್ ಮಿಷರ್, ಹಿ ಇಸ್ ದ ಫಸ್ಟ್ ಪರ್ಸನ್ ಟು ಐಸೋಲೇಟ್ ದಿಸ್ ಮಾಲಿಕ್ಯೂಲ್. ಐಸೋಲೇಟ್ ಅಂದ್ರೇನು? ನಾವು ಒಂದು ಸೆಲ್ ತಗೊಂಡ್ರೆ ವಿ ಹ್ಯಾವ್ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಇನ್ ಇಟ್. ಲೈಕ್ ಪ್ರೋಟೀನ್ಸ್ ಇದೆ, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್ ಇದೆ, ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ಸ್ ಇದೆ, ವೈಟಮಿನ್ಸ್ ಇದೆ, ಮೆನಿ ಅದರ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ ಆರ್ ದೇರ್. ಬಟ್ ಹಿ ಇಸ್ ದ ಒನ್ ಟು ಐಸೋಲೇಟ್. ಐಸೋಲೇಟ್ ಅಂದ್ರೆ ಇದೊಂದು ಮಾಲಿಕ್ಯೂಲ್ ನ ಸೆಪರೇಟ್ ಮಾಡ್ದ ಅವನು. Okay, so he just took the the molecule which is responsible for inheritance basically, and he called it nuclein anta karda. Yaar kardiddadna? Frederick Misha. Okay, Frederick Misher is the first person, okay, who actually found this in cells. wounded soldiers ಅಲ್ಲಿ ಸ್ವಾಬ್ ಅಂತ ಹಾಕಿರ್ತಾರೆ, ಅದರಲ್ಲಿ ಪಸ್ ಸೆಲ್ಸ್ ಇರುತ್ತೆ. ಆ ಪಸ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ he isolated this particular chemical compound. So as he isolated, from where he isolated, you know that there is, like, if you consider this as a cell has cytoplasm in it. In that there is one prominent structure which is present, which is called as a nucleus. So he isolated this material from nucleus of pus cells basically. Okay, and as he isolated from nucleus, he called this as nuclein anta karda. Okay, later discovery ಮಾಡ್ತಾ later they found out that this molecule is acidic in nature ಅಂತ ಕಂಡಿಡಿದ್ರು. ಅದನ್ನ ಕಂಡಿಡಿದಿದ್ದು ಆಲ್ಟ್ಮನ್. Then he named it as nucleic acid ಅಂತ ಕರ್ದ. Okay, nucleic acid. Nuclein ಅಂತ ಯಾಕೆ ಬಂದಿದ್ದು? Because it is present in the nucleus of the cell, so he called it as nuclein. Then they found out it is acidic in nature, so what did they call it? They called it nucleic acid. Okay, this is like just isolation of molecule. Idad mele, okay, after this, in 1869, okay, in 1869 all this research went on. ಇದೆಲ್ಲ ಆಗಿದ್ದು ಯಾವಾಗ? 1869 ಅಲ್ಲಿ. As there was no availability of such highly advanced scientific equipments and science was progressing during that time, it has taken a very long time to discover how this molecule is. But by 1953 we actually got the structure of this nucleic acid. Okay, how this nucleic acid is organized, that structure we got it in 1953. Okay, but this is not by one person. There are two people here who gave the structure of DNA. They are known as, one is James Watson. Okay, one is James Watson and another is, okay, already you might have heard about the names also, Francis Crick. Okay, Francis Crick. Okay, these two scientists gave the structure of the DNA. But ಈ ಸ್ಟ್ರಕ್ಚರ್ ಕೊಡಕ್ ಮುಂಚೆ ಇವರು ಜಸ್ಟ್ ಆ ಮಾಲಿಕ್ಯೂಲ್ ನ್ಯೂಕ್ಲಿನ್ ಇಂದ ಡೈರೆಕ್ಟ್ ಆಗಿ ಅವ್ರು ತಗೊಳ್ಳಿಲ್ಲ. ಇದ್ರ ಮಧ್ಯದಲ್ಲಿ ಎಷ್ಟೊಂದ್ ಜನ ಅದಕ್ಕೆ ಕಾಂಟ್ರಿಬ್ಯೂಟ್ ಮಾಡಿದ್ದಾರೆ. There are few scientists name which you should remember. One is Rosalind Franklin. And we also have Maury Wilkins. They did X-ray diffraction study. Idu tumba important experiment. Yenidu X-ray diffraction study? They passed X-ray through this molecule. Yav molecule ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದೀವಿ ನಾವು? DNA, deoxyribonucleic acid ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದೀವಿ. They passed the X-ray through this molecule. They were able to get an imprint where the light diffracted at an angle. With that, they were able to see in the core of the DNA, there is something present which tilts at an angle of around 35-degree angle alli tilt agide, core of the DNA nalli. That was given by Maury Wilkins and Rosalind Franklin. Okay, idadmele there is one more scientist name which is Shargaff. What he did, he took this nucleic acid material, separated all its component. Okay, ella adrallira components ella separate madbita, and then he gave, this is the percentage of a base present in it, this is the amount of sugar present in it. Like that, he gave the complete detail of the molecule. After studying all these things, finally, Watson and Crick gave us the model of DNA, that is deoxyribonucleic acid. Okay, when I started this lecture, I told you in living system we have something called nucleic acid. Okay, and this nucleic acid is of two category, I said. One is DNA, another is RNA, I said, right? Now, when you have to know about DNA, first you should know what is nucleic acid. ಹೌದಲ್ವಾ? ಫಸ್ಟ್ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಂದ್ರೆ ಏನು ಅಂತ ತಿಳ್ಕೊಂಡ್ರೆ then you can understand the structure of DNA. Okay, let us proceed further. So first ಏನ್ ತಿಳ್ಕೊಬೇಕು ನಾವು? First we should know what is nucleic acid, what it is made up of. Okay, nucleic acid is actually made up of monomeric units ಅಂತ ಕರಿತೀವಿ. Monomeric units ಅಂದ್ರೆ ಇವಾಗ ಒಂದು ಗೋಡೆ ಕಟ್ಟಬೇಕಾದ್ರೆ ಬ್ರಿಕ್ಸ್ ಇರ್ಲೇಬೇಕಲ್ವಾ? Bricks are the units in a wall, correct? Similarly, for nucleic acid, there is a bricks present, that is units present. ಯಾವ್ದದು small unit? That is called as nucleotides ಅಂತ ಕರಿತೀವಿ. Nucleotides ಅಂತ ಕರಿತೀವಿ. So, if many nucleotides ತುಂಬಾ nucleotide polymerize, okay, join ಆಯ್ತು ಅಂದ್ರೆ, then what do you call it? You call it one word add ಮಾಡ್ಬೇಕಿಲ್ಲ ಅಷ್ಟೇ, polynucleotides ಆಗತ್ತೆ. Many nucleotide when you add, they become polynucleotide. ಇವಾಗ ಇನ್ನೊಂದು ವರ್ಡ್ ಕಲ್ತ್ಕೊಂಡ್ರಿ, ಯಾವ್ದದು? ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಇದೆ. ಎಲ್ಲಿದೆ? ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ಸ್ ಅಲ್ಲಿ ಇದೆ. ಅಂದ್ರೆ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಒಂದೇ ಇದೆಯಾ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಲ್ಲಿ? ಇಲ್ಲ, ದೇರ್ ಆರ್ ಮೆನಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಪ್ರೆಸೆಂಟ್. ಓಕೆ, ಮೆನಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅಂದ್ರೆ ಈಚ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಲ್ಲಿ, every nucleotide, what it has, ಏನಿದೆ ಅದರಲ್ಲಿ. Okay, so when you take up nucleotide, this is made up of a compound which is called nucleoside. Okay, so ಈ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಲ್ಲಿ ಏನೇನೆಲ್ಲ ಇದೆ ಅಂತ ಫಸ್ಟ್ ಲಿಸ್ಟ್ ಮಾಡ್ಬಿಡೋಣ, ಆಮೇಲೆ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಬರ್ತೀನಿ. ಓಕೆ, ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಹ್ಯಾಸ್ ಫಸ್ಟ್ ಇನಾರ್ಗ್ಯಾನಿಕ್ ಫಾಸ್ಫೇಟ್ ಇದೆ ಇದರಲ್ಲಿ. ಓಕೆ, ಇನಾರ್ಗ್ಯಾನಿಕ್ ಫಾಸ್ಫೇಟ್. ಓಕೆ, ಒಂದೊಂದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಲ್ಲೂ ಇನಾರ್ಗ್ಯಾನಿಕ್ ಫಾಸ್ಫೇಟ್ ಇಟ್ ಹ್ಯಾಸ್. ಓಕೆ, ಅಂಡ್ ಇಟ್ ಹ್ಯಾಸ್ ಅ ನೈಟ್ರೋಜನ್ ಬೇಸ್. ಇಟ್ ಹ್ಯಾಸ್ ಅ ನೈಟ್ರೋಜನ್ ಬೇಸ್, ಒಂದು ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಕೂಡ ಇದೆ. ಪ್ಲಸ್ ಇಟ್ ಹ್ಯಾಸ್ ಅ ಶುಗರ್. ಯಾವ ತರ ಶುಗರ್? ಶುಗರ್ ಅಲ್ಲಿ ಬೇಸ್ಡ್ ಆನ್ ನಂಬರ್ ಆಫ್ ಕಾರ್ಬನ್, ನೀವು ಬಯೋಮಾಲಿಕ್ಯೂಲ್ಸ್ ಅಲ್ಲಿ ಓದಿದ್ದೀರಾ. ಇಫ್ ಶುಗರ್, ಸಿಂಪಲ್ ಶುಗರ್, if it has a 6 carbon, what do you call it? Can anybody answer? What do you call if it has a 6 carbon? I can see your answers. Okay, you can just tell me. Like if a simple sugar, if it has 6 carbon, ಏನಂತ ಕರಿತೀರಾ ಶುಗರ್ಗೆ? ನೀವು ಆಲ್ರೆಡಿ ರೆಸ್ಪಿರೇಷನ್ ಎಲ್ಲ ಓದಿರ್ಬೇಕಲ್ಲ. It is glucose ಬಗ್ಗೆ ಓದಿರ್ತೀರಾ. What do you call that sugar? Can you tell me? What do you call that sugar if it has 6 carbon in it? Hexose, very good. I think Renuka, I think, right? Very good. Okay, it is called hexose sugar. ಏನಂತ ಕರಿತೀವಿ? Hexose sugar ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, ಅದೇನೆ ಇವಾಗ 5 ಕಾರ್ಬನ್ ಇದ್ರೆ ಒಂದು ಸಿಂಪಲ್ ಶುಗರ್ ಅಲ್ಲಿ ಇಫ್ ಇಟ್ ಹ್ಯಾಸ್ 5 ಕಾರ್ಬನ್, what do you call it? What do you call it? Yes, it's called pentose. Very good. Yes, very good. It's called pentose sugar. Very good. Many people answered. Actually, I was not able to see the name. Lakshmi, I can see, and one more person who answered. Very good. Pentose sugar. Correct. So here in nucleotide, I'm not talking about poly. I'm talking about only nucleotide here. Okay. In each nucleotide, what and all component present? One inorganic phosphate. It has nitrogen base and it has a sugar, but the sugar which is present is a pentose sugar. A sugar which is present is a pentose sugar. Understood? Okay. Now let us take up how these are organized. ಅಂದ್ರೆ ಒಂದ್ರ ಪಕ್ಕ ಒಂದ್ ಇಡಕ್ ಆಗಲ್ಲ ಮಾಲಿಕ್ಯೂಲ್ ನ. It should be organized in a pattern. Correct? How it is organized? In order to understand, first you should know certain positions in pentose sugar. So first pentose sugar ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋಣ. Then we will go to nitrogen base. Then we will add a inorganic phosphate. Understood? Let us take up pentose sugar. When you take pentose sugar, what did I say? Pentose sugar has 5 carbon in it. A pentose sugar structure na ನಾನು ಈ ತರ ಬರಿಬೋದಲ್ವಾ? ಬಯೋಲಾಜಿಕಲಿ ಬರಿತಾ ಇದ್ದೀನಿ, ಕೆಮಿಕಲಿ ಕೆಮಿಸ್ಟ್ರಿ ಪೀಪಲ್ ನೋಡಿದ್ರೆ ನನಗೆ ಬೈತಾರ ಆಮೇಲೆ. ಓಕೆ, ಇಟ್ ಈಸ್ ಲೈಕ್ pentose sugar. I can write in this way, and you know that this is the carbon position. Okay, this is the carbon position, and you have a CH2OH group here. You have CH2OH group here, and there is H here, also OH here, also OH. H. You know that what is the formula of pentose sugar? Pentose C5. H. How much? How much? H. What is the general formula of simple sugar? Cn H2O N. Correct? No? So ಅವಾಗ H ಎಷ್ಟಿರ್ಬೇಕು? ಎಷ್ಟಿರ್ಬೇಕು? Right. How much? H 10. Must be there now. Very good. Okay. CH2OH H. This is the structure of pentose sugar. E pentose sugar ಅಲ್ಲಿ you need to remember this is the first carbon. Please remember this. This is the first carbon. This is second carbon position, third carbon position, fourth carbon position, fifth carbon position. Correct? ನಿಮ್ಗೆ ಕಾರ್ಬನ್ ಪೊಸಿಷನ್ ಕೂಡ ಜ್ಞಾಪಕ ಇರಬೇಕು, ಬಿಕಾಸ್ ಬೈಂಡಿಂಗ್ ಆಫ್ ಅದರ್ ಮಾಲಿಕ್ಯೂಲ್ is particular in nucleotide. Okay, this is the structure of pentose sugar. Okay, now let us take up nitrogen base. Okay, when you take up nitrogen base, there are 2 types of nitrogen bases present. What is that? One is called purine and another is called pyrimidine. Another is called as pyrimidine. Purine and pyrimidine ಎರಡಿದೆ. ಓಕೆ, ಪ್ಯೂರಿನ್ ಯಾವ್ದ್ಯಾವ್ದು? ಎರಡು ಪ್ಯೂರಿನ್ ಇದೆ. ಒಂದು ಅಡಿನೈನ್ ವಿಚ್ ಐ ರೆಪ್ರೆಸೆಂಟ್ ಆಸ್ ಎ, ಅನದರ್ ಗುವನೈನ್ ವಿಚ್ ಐ ರೆಪ್ರೆಸೆಂಟ್ ಆಸ್ ಜಿ. ಓಕೆ, ಈ ಪ್ಯೂರಿನ್ಗೆ ಪಿರಮಿಡಿನ್ಗೆ ಏನ್ ಡಿಫರೆನ್ಸ್ ಅಂದ್ರೆ, ಪ್ಯೂರಿನ್ ಈಸ್ a double ring structure. Purine is a double ring structure. ಈ ತರ double ring structure. Okay, while pyrimidine is a single ring structure. But in this ring, how many members are present? This is a 6-membered ring. This is a 6-membered ring. ಇಲ್ಲಿ ಏನು 9-membered ring. This is 9-membered ring. Okay, this is adenine also double ring structure. Guanine also double ring structure. But pyrimidine is single ring. Which are the pyrimidines present in living system? If you take up pyrimidines, we have thiamine, we have uracil, and we have cytosine. In living system, and when I said living system, you need to consider uracil also. But if I ask you DNA, then don't include uracil, because your DNA do not have uracil nitrogen base in it. Okay, so if you take up purines in nature, we have adenine, guanine, we have thiamine, uracil, cytosine. All these are the nitrogen base is present. ಅಂದ್ರೆ ಇದು ಯಾವ ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಬೇಕಾದ್ರೂ ಪೆಂಟೋಸ್ ಶುಗರ್ ಜೊತೆ ಕಂಬೈನ್ ಆಗಬಹುದು. Okay, but where does it combine? How does it combine? Whenever this sugar binds to pentose, whenever this nitrogen base binds to pentose sugar, it will always bind to the first carbon OH group. Please remember, it will bind to first carbon OH group. First carbon OH group ge ಯಾವ್ದು bind ಆಗುತ್ತೆ? Nitrogen base will bind. Okay, now it can bind to any. Adenine can bind here, guanine can bind, cytosine can bind, thymine can bind. Okay, but here one difference ide. ಏನು difference? ಇದು ribose sugar. Okay, this is ribose. How do you know it is ribose? When you find there is like two OH group, that is in the second carbon position OH group is present. In the third carbon group also OH group is present. Then it is having a completely OH group, no, nothing is missing here. We call it as a ribose sugar. Okay, but ಇದೇ sugar na ನಾನು ಹಿಂಗೆ ಬರ್ದೆ ಅನ್ಕೊಳ್ಳೋಣ. Okay, I will not write in detail, I will just write here. Here instead of writing OH, I am writing H here, here OH. Suppose if I write in this way, then there is one oxygen less in this, correct? When one oxygen is less, what do you call this sugar as? This is called deoxy. Why did you call it deoxy? Because there is one oxygen less. So it is called deoxyribose. This is deoxyribose. Then what is this sugar? This is ribose sugar. So what is the difference between deoxyribose and ribose? It is the OH group present in the second carbon is the indication of ribose. The H group of the second carbon is the indication of deoxyribose. So in DNA expand ಮಾಡ್ಬೇಕಾದ್ರೆ ನೀವೇನ್ ಹೇಳಿದ್ರಿ? Deoxyribose sugar, you said, right? So when you said deoxyribose sugar in DNA, which sugar is present? It is this sugar, deoxyribose sugar, which is present. You don't find ribose sugar in DNA. Then where do you find ribose sugar? In RNA, you will find ribose sugar. In RNA, you will find ribose sugar. Okay, understood. Now you know about pentose sugar. There are two categories. But when they ask you which is the sugar present in DNA, you must select deoxyribose sugar. Which is the sugar present in RNA? It is ribose sugar. Done? Okay. Now come to nitrogen bases. Okay, when you come to nitrogen bases, there are two categories, one purine or pyrimidine. Okay, but in case of DNA, we have adenine, okay, we have guanine, we have thymine and cytosine. So associated with DNA, nitrogen bases associated with DNA is adenine, thymine, guanine, cytosine. Okay, A, T, G, C. All these are the nitrogen bases associated with DNA. But with RNA, okay, if you take up RNA, with RNA, you will find adenine, but no thymine. Instead of thymine, you will find uracil and you will find guanine and cytosine. AUGC is in RNA. Okay, now let us see how nitrogen base links to the pentose sugar. How nitrogen base links to pentose sugar. First ಅದು ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕಲ್ಲ. So first this is like pentose sugar. Okay, this pentose sugar, this is the first carbon position, correct? To this first carbon position, a nitrogen base links. Okay, which is this nitrogen base? Let us consider this as adenine. Okay, now this is called a sugar bound with a nitrogen base with the help of a bond, glycosidic bond. Okay, N-glycosidic bond we say. Why did you say N? Because in the double membered ring, which is the double membered ring? Purine is the double membered ring. In that double membered ring, there is nitrogen present. So as this bond is between carbon and nitrogen, We call it as N-glycosidic linkage. ಏನಂತ ಕರಿತೀವಿ? N-glycosidic linkage. Now, this molecule which is formed, we call this as nucleoside ಅಂತ ಕರಿತೀವಿ. It is called as nucleoside. Okay, ನಮಗ್ ಬೇಕಾಗಿರೋದು ಯಾವುದು? Nucleotide. But what did we get? We get nucleoside. Whenever pentose sugar binds with adenine or any nitrogen base. ಇವಾಗ ನಾನು ಹಿಂಗೊಂದು pentose sugar ಬರ್ದೆ. Okay, ಈ pentose sugar ಅಲ್ಲಿ ಇದು consider it is H, that is deoxyribose. ಇದಕ್ಕೆ I can link this OH to another T ಕೂಡ ಇರ್ಬೋದು. What is that? Thymine can bind. But whenever a pentose sugar binds to nitrogen base, we call this nucleoside. Okay, but nucleoside type ಬೇರೆ ಬೇರೆ ಆಯ್ತು. ಅವಾಗ ಅಡಿನೈನ್ ಬೈಂಡ್ ಆಗಿದ್ರೆ what do you call this nucleoside? It is called adenosine ಅಂತ ಕರಿತೀವಿ. ಅಡಿನೈನ್ ಬೈಂಡ್ ಆದ್ರೆ adenosine. ಥೈಮಿನ್ ಬೈಂಡ್ ಆದ್ರೆ ಥೈಮಿಡಿನ್. ಥೈಮಿನ್ ಬೈಂಡ್ ಆದ್ರೆ ಥೈಮಿಡಿನ್. Did you understand? So, when you take up a sugar and a nitrogen base, nucleoside. But what do we want? We want a nucleotide. So, when you take up the phosphate moiety, I told you, inorganic phosphate is the third component in a nucleotide. So, when you take up a phosphate, let me represent a phosphate in this way, P. This phosphate will always bind to the fifth OH group. Fifth OH group andre fifth carbon OH group, that is outside. Okay, so whenever a phosphate will bind to a nucleoside, idu nucleoside, correct? This is nucleoside. Now this is not nucleoside. Why this is not nucleoside? Because phosphate is bound to a nucleoside. Then this becomes thymidine, which was earlier. Now as phosphate has bound, it will become thymidilic acid ಆಗ್ಬಿಡತ್ತೆ. Thymidilic acid ಆಯ್ತು. Or you can also say thymine or thymidine monophosphate ಅಂತ ಕೂಡ ಹೇಳ್ಬೋದು. monophosphate. ಯಾಕೆ monophosphate ಅಂತ ಕರಿತಾ ಇದ್ದೀರಾ? Because there is one phosphate associated with it. ಇವಾಗ ಇದೇನಾಯ್ತು? This becomes a nucleotide. It becomes a nucleotide. Now come to nucleic acid. Such nucleotide polymerized to become polynucleotide. Such nucleotide many polymerize, tumba polymerize. Then it becomes a polynucleotide. Okay, ಇವಾಗ polynucleotide ಜೊತೆಗೇನೆ DNA structure ಕೂಡ ಒಟ್ಟೊಟ್ಟಿಗೆ ಕಲ್ತ್ಕೊಂಡ್ಬಿಡೋಣ. It will be easy for you. Okay, so let us take up a polynucleotide and the structure of DNA. Why DNA here? Because DNA is a double stranded structure. ಈ ಡಬಲ್ ಸ್ಟ್ರಾಂಡ್ ಅಂತೀವಲ್ಲ, ಅಂದ್ರೆ ಒಂದ್ ಸ್ಟ್ರಾಂಡ್ ಅಲ್ಲಿ ಏನಿದೆ, ಈಚ್ ಸ್ಟ್ರಾಂಡ್ ಇಸ್ ಅ ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್. ಅಂದ್ರೆ ಎರಡು ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ನ ಜಾಯಿನ್ ಮಾಡಿದ್ರೆ ದೆನ್ ಇಟ್ ವಿಲ್ ಬಿ ಎ ಡಬಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ DNA. Okay, two polynucleotide chain combined is one double stranded DNA. ಅರ್ಥ ಆಯ್ತಾ? So now let us take up a single polynucleotide. When you take a single polynucleotide, I will be writing a pentose sugar and a phosphate in this way. Just a graphical representation for you to understand ಅಷ್ಟೇನೆ. So ಇಲ್ಲಿ ನಾನು ಎಷ್ಟು ಬರ್ದಿದ್ದೀನಿ? 3 ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ನ ಬರ್ದಿದ್ದೀನಿ. Is it nucleotide? Can anybody say yes or no very quickly? Is it a nucleotide which I have written there? Is it a nucleotide? No, very good. It's not. It's not a nucleotide. Why? ಏನ್ ಇರ್ಬೇಕು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಅಲ್ಲಿ? 3 ಕಾಂಪೊನೆಂಟ್ ಇರ್ಬೇಕು ಅಂದೆ. I have only one pentose sugar and phosphate. ಎರಡೇ ಇದೆ. ಇನ್ನೊಂದು ಕಾಂಪೊನೆಂಟ್ ಯಾವುದು? We should write what? Nitrogen base ಅಲ್ವಾ? Okay. So nitrogen base I will represent in this way. ಇವಾಗ ಎ ಬರ್ದೆ ಅಂದ್ರೆ this is what? Adenosine monophosphate ಆಯ್ತು. ಅದನ್ನೇ ಇಲ್ಲಿ ಬಂದು ನಾನು ಜಿ ಬರಿತೀನಿ. G is also double membered ring, no? So double ring structure, G. This is guanosine monophosphate ಆಯ್ತು. Okay, next come to ಇಲ್ಲೊಂದು ಬರೆಯೋಣ. ಇದನ್ನೇನ್ ಬರೆಯೋಣ? T ಬರೆಯೋಣ. Okay, that is thymidine monophosphate. ಇನ್ನೊಂದು ಯಾವುದು DNA ಇರೋದು? Can anybody tell me ಇನ್ನೊಂದು ಯಾವುದು? ಶುಗರ್ ಬೇಗ. ನೈಟ್ರೋಜನ್ ಬೇಸ್ ಯಾವುದು? ಇನ್ನೊಂದೇ ಒಂದು ಬಿಟ್ಬಿಟ್ಟಿದ್ದೀನಿ. ಯಾವ್ದದ್ದು ನೈಟ್ರೋಜನ್ ಬೇಸ್ ವಿಚ್ ಇಸ್ ದೇರ್ ಇನ್ DNA? Very good. Cytosine. Who is that? Biradar. Okay, very good. Cytosine. Correct. So let me write this here. Cytosine. Very good. Okay. So, ಅಂದ್ರೆ ಒಂದು DNA ನಲ್ಲಿ ಬರೀ 4 ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇರುತ್ತಾ? ಹಲೋ ಇಲ್ಲ. It is a polynucleotide. Thousands and thousands of molecules will be there. Thousands ಏನು 10 to the power of -9 ಅದದ್ದು ಲೆಂತ್ ಬರುತ್ತೆ. ಅಂದ್ರೆ I mean millions you can say. So many base pairs will be there in it. ಬರೀ 4 ಇರಲ್ಲ. But combination ಈ ತರ ಇರುತ್ತೆ. Combination will be between I mean like A, T, G, C ನೇ ರಿಪೀಟ್ ಆಗಿರ್ಬೋದು. ಯಾವ ತರ ರಿಪೀಟ್ ಆಗ್ಬೋದು? ಎ ಒಂದ್ 10 ಇರ್ಬೋದು, ಜಿ ಒಂದ್ 10, ಟಿ ಒಂದ್ 10. ಸೊ ಕಾಂಬಿನೇಷನ್ ಬೇರೆ ಬೇರೆ. ಬಟ್ ಅದ್ರಲ್ಲಿ ಇರೋಂತ 4 ನೈಟ್ರೋಜನ್ ಬೇಸಸ್ ಯಾವ್ದ್ಯಾವ್ದು? ಎ, ಟಿ, ಜಿ, ಸಿ. ಇದು 4 ನೈಟ್ರೋಜನ್ ಬೇಸಸ್. Did you understand? Now, idu, if I have represented here, have I represented polynucleotide here or have I represented many nucleotides? ಎಷ್ಟು ಡಿಫರೆನ್ಸ್ ಇದೆ? ಮೆನಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಇದೆಯಾ ಇಲ್ಲಿ, ಇಲ್ಲ ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇದೆಯಾ ಇಲ್ಲಿ? ಎರಡರಲ್ಲಿ ಒಂದು ಆಪ್ಷನ್ ಕ್ವಿಕ್ ಆಗಿ ಅಂತ ಹೇಳ್ಬಿಡ್ಬೇಕು. ಮೆನಿ ನ್ಯೂಕ್ಲಿಯೋಟೈಡಾ ಪಾಲಿನ್ಯೂಕ್ಲಿಯೋಟೈಡಾ? ಪಾಲಿ ಅಂದ ತಕ್ಷಣನೇ ಅದು ಲಿಂಕ್ ಆಗಿರ್ಬೇಕಲ್ವಾ? ಕರೆಕ್ಟ್? ಇಸ್ ಇಟ್ ಲಿಂಕ್ಡ್? ಎಸ್ ಆರ್ ನೋ? ಕ್ವಿಕ್ಲಿ. ಇಸ್ ಇಟ್ ಲಿಂಕ್ಡ್? No, it's not linked. Correct? So very correct, very correct, very correct, Jyote. So it is not linked. So I have written many nucleotides here, but I have not joined them. So when you join, okay, how do you join? Please remember the bond here. A phosphate of one nucleotide always binds to, okay, where is the phosphate position? In the 5th carbon position, correct? The 5th carbon phosphate position of one nucleotide binds to the OH group of the 3rd carbon position of the next or the neighboring nucleotide. Okay, so ee tara if they link, always the link is in this way, then now the bond which is present, iವಾಗ ಇದು first one ತಗೊಳ್ಳೋಣ. The bond which is present between sugar and a phosphate is called ester bond ಅಂತ ಕರಿತೀವಿ. This is called ester bond. Okay, but as there are two ester bond present in a polynucleotide linking 2 nucleotides each, we call it as phosphodiester bond. It is called phosphodiester bond anta kariteve. Okay, now is this one polynucleotide? Yes. Now we know which is polynucleotide. That is, this is one polynucleotide. In this polynucleotide, yenella bonds banto. You need to remember, MCQs will be framed on that. One is glycosidic bond, which is linking sugar and nitrogen base. Another is ester bond, which is linking a nucleoside with a phosphate. Phosphodiester bond linking one nucleotide to the other nucleotide of the same chain. Of the same chain. Now you understood. This is one nucleotide chain, polynucleotide chain. Ivaga, now we'll come to DNA structure. Okay, when you come to DNA structure, DNA, we usually say it is a double helix anta karite. It is a double helix. Yake double annadu? Because it is not made up of one polynucleotide. It is made up of many, okay, nucleotide sequence. It is made up of many nucleotide sequence. Okay, so this nucleotide sequence which is I mean present, okay, which is present is basically ಈ ತರನೇ ಬರೆಯೋದು. DNA structure ಬರಿಬೇಕಾದ್ರೆ ಆಮೇಲೆ ಸ್ವಲ್ಪ modify ಮಾಡ್ಕೋತೀನಿ. Okay, lightly I'll modify. Okay, here when you take up, there is, okay, let me write that. When you take up the DNA, there are two nucleotide sequences. Okay, one is this one. Okay, this is the sequence which is present here. Okay, this is the nucleotide sequence. Okay, this is the backbone of the DNA. ಇದನ್ನೇನಂತ ಕರಿತೀವಿ? Backbone of the DNA ಅಂತ ಕರಿತೀವಿ. Okay, this backbone of the DNA, please remember, backbone of the DNA is made up of, okay, ಇದ್ರ ಮಧ್ಯದಲ್ಲಿ ಕೆಲವು ಬಾಂಡ್ಸ್ ಇರುತ್ತೆ ಹಿಂಗೆ. Okay, these, I mean, backbone of DNA is made up of sugar and phosphate. Backbone of DNA is made up of sugar and phosphate. It is made up of sugar and phosphate. Sugar and phosphate ಎಲ್ಲಿ ಬರುತ್ತೆ? Backbone of DNA ನಲ್ಲಿ ಬರುತ್ತೆ. Okay, it is made up of sugar and phosphate. ಅಂದ್ರೆ ಈ backbone ಅಲ್ಲಿ ಏನಿದೆ? Sugar and phosphate. What is here? Sugar. Which is the sugar present in DNA? It is deoxyribose sugar. Sugar and phosphate is present in the backbone of DNA. Sugar and phosphate is present in the backbone of DNA. Okay, DNA backbone. Idu backbone. Okay, it is present in the sugar and phosphate is present in the backbone of DNA. ಅಂದ್ರೆ ನಾ ಬರ್ದಿರೋ ಬ್ಯಾಕ್ ಬೋನ್ ಬಂದು ಇಲ್ಲಿಂದ ಇಲ್ಲಿವರೆಗೆ ಮಾತ್ರ. ಇಟ್ ಇಸ್ ಟ್ವಿಸ್ಟೆಡ್. Okay, it is twisted like that. Then what is there in the central part of the DNA? In the central part of the DNA, one side it has till here, it has nitrogen bases present. Okay, nitrogen bases ಇದೆ. ಇದು ಒಂದ್ ಕಡೆದು nitrogen bases. That means adenine, guanine, thymine, cytosine, which I have represented, that is the nitrogen bases protruding to the core of the DNA. Core of the DNA ಏನಿದೆ? Nitrogen bases are present. To the core of the DNA, there is nitrogen bases present. ಆಯ್ತಾ? But ಇದಕ್ಕೆ opposite strand on ಬರುದ್ನಲ್ಲ. So here also I should write the opposite strand, right? So let us write that. So opposite strand also has, same pentose sugar. Okay, it has pentose sugar, and you know that nucleotide has pentose sugar, and it has a nitrogen base, and it has a phosphate. Backbone is, you know that it is made up of a phosphate. Okay, it is made up of phosphate. And what is the nitrogen base present here? It is usually thymine. Okay, it is usually cytosine. Hello, why am I writing like in particular? In particular, I am writing in this way because Shergaff, I discussed about one scientist's name, right? Shergaff. What he did analyze that, the amount of adenine present in DNA is equal to or same as that of thymine present. Okay, the amount of adenine present in DNA. Okay, let me write that. The amount of adenine present in DNA is equal to the amount of thymine present. ಹಂಗಂದ್ರೆ ಏನರ್ಥ? Adenine will bind to thymine. ಅದಕ್ಕೆ ಅದೆರಡು equal amount ಅಲ್ಲಿ ಇರುತ್ತೆ. ಕರೆಕ್ಟಾ? So whenever in one of the nucleotide adenine is present, the opposite chain of nucleotide will have thymine, and it is linked by a special type of bond between these two. This bond which connects these two is called hydrogen bond. This is the hydrogen bond. Now adenine of one polynucleotide chain binding with thymine of the other polynucleotide, now it is polynucleotide chain, is known as a base pair. ಇದನ್ನೇನಂತ ಕರಿತೀವಿ? This is called as a base pair. This is known as a base pair. Please remember, I'll repeat. One nitrogen base of one polynucleotide linking to the other nitrogen base of the opposite nucleotide, we call this as base pair. But what is there in the backbone of the DNA, whether it is the one backbone of polynucleotide chain or the other, it has only sugar and phosphate in it. Then what is there in the core of the DNA? In the core of the DNA, we have base pairs present. Base pair henge one nitrogen base of one nucleotide, opposite base pair of the other nucleotide, both combined with the help of a bond, which is that hydrogen bond. Okay, so this is a base pair present. So that is why it acts like the staircase. Okay, the core of the DNA is like staircase. So what is this here staircase formed of? It is the base pairs which are there in the core of the DNA. Did you understand? And remember, always adenine to thymine. How many hydrogen bond will be present between adenine to thymine? Only 2 hydrogen bond. But it is not the same case with the guanine and thymine, guanine and cytosine. It has 3 hydrogen bonds present here. So this is another base pair. This is one more base pair. So G and C, between G and C, G represents guanine, C represents cytosine. So between G and C, how many hydrogen bonds present? 3 hydrogen bonds present. Did you understand? So a DNA is formed by combining 2 polynucleotide chains. Okay, 2 polynucleotide chain. Okay, now based on this, we can frame many questions in MCQ. Consider a DNA is 100 base pairs. A DNA has 100 base pairs, just for calculation purpose. Suppose if I tell you in this 100 base pairs, I'll give you two questions here. One question I'll ask, if out of 100 base pair, if this DNA has 10 adenine, if this DNA has 10 adenine, then how many thymine is present anta keldre. What is the answer? How many thymine is present anta keldre. 100 base pair DNA. Very good, Jyoti. It is 10 only. Very good. Okay. Now I will change the question. Same DNA. 100 base pair DNA. In the 10 adenine, good. Now I will ask how many cytosine present. How many cytosine present? Now what you will answer? Let us see what you will answer. Jyoti, you are right with thymine. Very good. But now I changed the question. What is the question? If in 100 base pair long DNA, if 10 adenine present, then how many cytosine? Saran. Okay, I can't spell the whole name. Spurti also answered, I believe. Very good. Okay, it is. How much? Any other? Any other trials here? Okay, very good. Your answer is right. How much? Let us calculate how much. Okay, if adenine is 10, then thymine is, one of your friend told it is 10. So totally, thymine is also 10. So totally how much is over now? Nitrogen bases 20 over. Out of how much? 100. 100 base pairs I said. That means 20 is over here. Correct? No? Remaining how much in 100 base pair? Remaining 90. If you, divide it between G and C, okay, between G and C, if you divide it, 40 will be G, 40 will be C, because GC only will pair. Okay, now the question is, when I told 100 base pair, you can also consider it as 200 bases, correct? Yes or no? 100 base pair, when I said, you can also consider it as 200 bases, correct? Is it not? Yes or not? Am I correct or wrong? You can consider it as 200 bases, correct? Okay, out of 200 bases, how many I said adenine is present? How many I said adenine is present? This is what we call discussion. Basically, you need to answer. Okay. So actually you people calculated. because 100 base pair I have given. So you said 10 adenine means 10 will be, thymine. So totally 20. Then remaining 40 you calculated in that way. Correct? No. But actually what I have given here, base pair I have given. When I have given base pair, then 200 bases. Correct? No. Out of 200 bases, how many adenine? 10 adenine, correct. If 10 adenine is present, how many thymine? 10 thymine will be present, correct. Okay, so totally, okay, 20 bases over. So out of 220 bases over, how much is remaining? Yes, 20 bases over here. So how much is remaining? 100 and? Yes. Very correct. Very correct. Who is that? Rathod, Jairaj. Very good. So how much is remaining? 180 bases are remaining. Correct? No. Okay. Then out of 180 bases, half of it. How much? How much? Half of it. Yes. You know maths better than me. Okay. 90. Okay. So 90 bases will be of what? G. 90 bases will be of C. Correct, right? Is it not? So which answer is correct? Is it 40 base correct, which you answered earlier, or is it 90 base correct? Which is correct? Hello. Which is correct? Yes, Renuka, you are right. 90. Very correct. Jairaj, 90 correct now? See how we do mistakes. Okay. Now, if I had asked you a question for the same, like 100 base pair, listen carefully, 100 base pair long DNA. Okay, we'll take the same base pair long DNA. Suppose instead of saying nucleotide or nitrogen base, if I give you 10 or 80 base pairs. See the question now. What did I change? I just changed one small unit there. What did I do? Like 100 base pair long DNA. Out of that 10 are 80 base pairs, I said. 1080 base pairs over. Out of 100 base pair, 1080 base pairs are over. Then how many will be, if I ask you, how many will be GC base pairs? Now what will be the answer? Now what will be the answer? How many will be GC base pair? 30 you should answer. You know that. You know that. Very correctly you know that. I have changed another question here. 100 base pair long DNA. Right. Out of that I said 10 or 80 base pairs. Then how many are GC base pairs? 90. Very correct. I think Rathod you answered now. Very good. 90 are 80 GC base pairs. Correct. Okay. So please be careful when you are answering MCQs. Have they given base pairs? Have they given bases? Or have they given asked you to find out base pairs? Check with the question. Okay. If it is based on bases, then you should convert the base pair to bases, number of bases. Did you understand? Okay. But I can frame questions in different way also. Okay. Like, for example, if you take this DNA, okay, in this DNA, this is the backbone of DNA and there is something called minor groove. This is called minor groove. Okay. And this is called major groove. Okay. Major groove. Basically, what is this groove is, it is just the turn of the DNA. Why the DNA is turning? Because of molecular bonds present in them. It is turning at an angle of 35 degrees. Okay. So that is why it is like a helix. It is like a helix. What type of helix? It is a right-handed helix. DNA is a right-handed. It is a right-handed helix. It is a right-handed helical structure. As it is turning, there will be one major turn and one minor turn. As it is turning, there will be one major turn and one minor turn. Okay. So because of that, there is like, we call it as minor groove and major groove, we say. Okay. Now, when you take out one complete turn of the DNA, when you take one complete turn of the DNA, it is called as one pitch, we say. It is known as one pitch. Okay. Now, one pitch is equal to base pairs. One pitch is equal to 10 base pairs. So I can frame question based on pitch also. If in a pitch there are 3 adenine present, if in a pitch of DNA 3 adenine molecule present, then how many? Okay, how many? Thymine, easy. 3 adenine means 3 thymine, done with it. But how many G present if I ask, then please remember it is 10 base pairs. Okay, based on that you calculate. Did you understand? Is it clear to you? Yes or no? Is it clear? Right, very good, very good. Okay, so based on pitch also I can give you calculations. Done. So when you take up a DNA, please remember it is a double helix. You understood why it is a double helix, because there is 2 polynucleotide chain is present. One. Second, the backbone of the DNA is made up of sugar and a phosphate. Okay, 2. 3. The core of the DNA is made up of nitrogen base pairs. Core of the DNA is made up of pairs, base pairs. The distance between 2 base pair, this base and this base pair, the distance between 2 base pair is 0.34 nanometer. Nanometer, when I say, it is equal to 10 to the power of minus 9 meters. Remember the length here. When I say 0.34 nanometer, then the length is here, only this length is 10 to the power of 9 meters. That is a billionth of a meter. Billionth of a meter is that small distance here, 0.34 nanometer. Did you understand? Now, when you take up, okay, this is regarding the distance between base pair. Okay, now remember the DNA is lying in opposite manner. Why it is lying in opposite manner? Which is this end, this phosphate which is present is attached to the 5th carbon of the pentose sugar, correct? So 5th OH group, correct? So this end is called 5' end of the DNA. You see the other end, which end is open? 3 carbon position is open, so we call it as 3 OH group is open, so we call it as 3' end of DNA. You take up the opposite side. Now this is 3' and this is 5' of the DNA. So always in DNA the polynucleotides are anti-parallel in nature. It is anti-parallel in nature. Okay, now I told you the distance here. I told you basic structure of DNA, not in detail, the basic structure of DNA. Now, when you know that distance between 2 base pair is 0.34 nanometer, then in human body, okay, in human body, we have, like if you take the complete DNA diploid set, it is 6.6 into 10 to the power of 9 base pairs present. If it is 6.6 into 10 to the power of 9 base pair, then what is the length of the DNA? I can ask length also, no? In meters. Then how will you calculate? If it is 6.6 into 10 to the power of 9 base pairs is the, number of bases present in one cell of human body. Okay, one cell, single cell. Then what is the distance between 2 base pair? What is the distance length? It is 0.34. Much? 0.34 into 10 to the power of minus 9 meters, right? 10 to the power of minus 9 meters, correct? Okay, now if you convert this to meters, then how much will be? Like multiply 6.6 into 0.34. It is around 2. meters. Okay, that means such a long DNA, 2-meter long DNA is compactly arranged in micron size nucleus. It is compactly arranged in micron size nucleus. Okay, this arrangement of that such a long DNA into compactly arranged structure, we call it as DNA packaging. It is called as DNA packaging. How this DNA is packed? Okay, that is like really interesting story here. It is like whenever somebody gives you a bundle of real threads, threads, when they give you, if they ask you to separate based on the color, what you will do, you will take a stick and start winding it around it. Correct? No, it will reduce your work of segregating different colored threads. Correct? Okay, same thing the cell also did. What it will do, it will wind it around, but there is no stick there. Instead of stick, it will use a special type of protein. Which is that protein? It is called histone protein. Okay, so when this long DNA, okay, this is the DNA structure. Now this DNA is now compactly arranged in, okay, with the help of special type of protein in eukaryotes, okay, which is that histone proteins. When you take this, I can write this DNA, like to simplify, I'll write this DNA like this. Now this DNA, double-stranded DNA, is coiled around a special type of protein which is called as histone protein, which is known as histone protein. Okay, and in histone protein we have two types here. One, okay, there are, I mean, 2H2A histone, okay, and we have 2H2B histone, we have 2H3, and we have 2H4. So when all of them are present in double number, so how many histone proteins will be there here? There will be 8 histone proteins which will be there here, which is that 2 times H2A, 2 times H2B, 2 times H3, 2 times H4. Totally how many? 8 of them. So we call this as histone octamer. We call it as histone octamer. Okay, now around this histone octamer, there will be the DNA which will coil around itself. It will coil around itself like this. Okay, so this is the histone octamer. Now this DNA is coiling around the histone protein. Okay, and there is one linker histone present. This linker histone is called H1. H1 is not part of the core histone protein. Histone octamer has only these, but H1 is the peripheral histone which is present. This one whole structure, we call it as nucleosome. It is called as nucleosome. Okay, in nucleosome, what you find? You find histone protein. You find double-stranded DNA, which is coiled around histone octamer. This is called nucleosome. Suppose if I write many nucleosome like this, you will find that DNA will be forming a beads on string like structure. DNA will be coiled around histone protein like a beads on string. We call this structure as chromatin. Okay, this is called chromatin.
ಎಷ್ಟೊತ್ತಾಗುತ್ತೆ ಮೇಡಮ್?
ಸರ್ ಇನ್ನೊಂದು 2 minutes ಸ್ಟಾಪ್ ಮಾಡ್ಬಿಡ್ತೀನಿ.
ಅಲ್ಲಲ್ಲ, ಅಕಸ್ಮಾತ್ ಏನಾದ್ರು ಇನ್ನೊಂದು ಕ್ಲಾಸ್ ಏನಾದ್ರು ಕಂಟಿನ್ಯೂ ಆಗೋಂಗಿದ್ರೆ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಮಾಡ್ಲಿ ಅಂತ ಅನ್ಕೊಂಡಿದ್ದೆ. ಮಾಡಿ ಮೇಡಮ್, ತೊಂದರೆ ಇಲ್ಲ.
ಇನ್ನೊಂದು ಕ್ಲಾಸ್ ಇವಾಗ್ಲೇ ತಗೋಬೋದಾ ಸರ್?
ಇಲ್ಲ ಇಲ್ಲ ಇಲ್ಲ, ಸಾರಿ.
ಇಲ್ಲ,
ಮನೆಗೆ ಹೋಗುವಂತ ಇದ್ರಲ್ಲಿ ಇರ್ತಾರೆ.
Okay.
ನೆಕ್ಸ್ಟ್ ಟೈಮ್
I will continue from here in the next class, sir. That is also fine with me because there is so much to say.
ಕಂಟಿನ್ಯೂಯೇಷನ್ ಇದ್ರೆ ನೆಕ್ಸ್ಟ್ ಒಂದು ಕ್ಲಾಸ್ಗೆ
ಹಾ,
ನಾನೊಂದು ಡೇಟ್ ನಿಮಗೆ ಪ್ರಿಫರ್ ಮಾಡ್ಕೊಡ್ತೀನಿ ಮೇಡಮ್.
ಶೋರ್ ಸರ್, ಶೋರ್. ಕಂಟಿನ್ಯೂಯೇಷನ್ ಇದೆ ಇಲ್ಲಿಂದ, ಯಾಕಂದ್ರೆ ಇನ್ನ ಪ್ಯಾಕೇಜಿಂಗ್ ಬಂದಿಲ್ಲ ನಾನು.
ಒಂದು ಲೈನ್ ಎಂಡ್ ಮಾಡಿ ಮೇಡಮ್, ಆಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಕಂಟಿನ್ಯೂ ಮಾಡೋಣ.
Sure, sir. I'll end in one point. Okay. After that,
Please,
you can
please.
stop. Yeah. Right. So what is happening here? This double-stranded DNA is coiling around histone protein, and it is forming nucleosome, and such nucleosome, you can see here, such nucleosome, it is forming a thread, okay, like beads on string ತರ ಕಾಣಿಸ್ತಾ ಇದೆಯಲ್ಲ. So we call this as a chromatin. Okay, but this chromatin won't stay like this. It will further coil upon itself. Okay, and when it coils upon itself, here and there, ಇದೇ DNA ತಗೊಳ್ಳೋಣ, here and there it might be coiled more, and then again beads on string, again coiled more beads on string, because 2.2 meter long DNA ನ split ಮಾಡ್ಬೇಕಲ್ವಾ ನಾವು, condense ಮಾಡ್ಬೇಕಲ್ಲ. So what we see is, here and there you will see dense regions of like packed, highly packed DNA present. We call this as heterochromatin ಅಂತ ಕರಿತೀವಿ. ಹೆಟರೋಕ್ರೊಮ್ಯಾಟಿನ್ ಅಂತ ಕರಿತೀವಿ. ಬಟ್ ಅದೇ ಲೂಸ್ ಆಗಿ ಪ್ಯಾಕ್ ಆಗಿದೆಯಲ್ಲ, ಅಂದ್ರೆ ಬರೀ ಬೀಡ್ಸ್ ಆನ್ ಸ್ಟ್ರಿಂಗ್ ತರ, ಬರೀ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಸಪರೇಟ್ ಆಗಿ ಲಿಂಕ್ ಆಗಿ ಒಂದು ಬೀಡ್ಸ್ ಆನ್ ಸ್ಟ್ರಿಂಗ್ ತರ ಕಾಣಿಸ್ತಿದ್ಯಲ್ಲ, ವಿ ಕಾಲ್ ದಿಸ್ ಆಸ್ ಯುಕ್ರೋಮ್ಯಾಟಿನ್ ಅಂತ ಕರಿತೀವಿ. ಯುಕ್ರೋಮ್ಯಾಟಿನ್. ಓಕೆ, ಹೆಟರೋಕ್ರೊಮ್ಯಾಟಿನ್ ಏನೋ ಡೆನ್ಸ್ಲಿ ಪ್ಯಾಕ್ಡ್ ಏರಿಯಾ, ಯುಕ್ರೋಮ್ಯಾಟಿನ್ ಏನೋ ಲೈಟ್ಲಿ ಪ್ಯಾಕ್ಡ್ ಏರಿಯಾ. Okay, a lightly packed area is highly active. In activity, protein synthesis ge it will help. Okay, it will help in protein synthesis. That is why euchromatin is highly active region. I mean transcriptionally active region, but heterochromatin is packed area. It is not highly active. Okay, ಈ ತರ chromatin ಮತ್ತೆ ಕಾಯಿಲ್ ಆಗಿ ಏನಾಗುತ್ತೆ, ಫರ್ದರ್ when there is too much of coiling taking place, finally it will lead to what do we call it as, like the formation of one double stranded DNA. ಒಂದು double stranded DNA will be highly compactly arranged into specialized structure. Which is the specialized structure? This is called as chromosome. ಅದನ್ನ ನಾವು ಹಿಂಗ್ ಬರ್ದ್ಬಿಡ್ತೀವಿ ಈಸಿಯಾಗಿ, ಕ್ರೋಮೋಸೋಮ್ ಗೆ ಫಾರ್ಮ್ ಆಗತ್ತೆ. ಆದ್ರೆ ಕ್ರೋಮೋಸೋಮ್ ಫಾರ್ಮ್ ಆಗ್ಬೇಕಾದ್ರೆ, when it has to become a chromosome, it has to bind with ಬರೀ ಹಿಸ್ಟೋನ್ ಪ್ರೋಟೀನ್ ಜೊತೆ ಆಗಲ್ಲ, ಸಾಕಾಗಲ್ಲ. Along with histone protein, it will also bind with other protein complexes, which is called as non-histone protein complexes. Non-histone, histone ಅಲ್ಲ, histone ಬಿಟ್ಟು ಬೇರೆ ಪ್ರೋಟೀನ್ ಕಾಂಪ್ಲೆಕ್ಸಸ್ ಜೊತೆ ಕಾಂಪ್ಯಾಕ್ಟ್ ಆಗಿ it is highly condensed. It is highly condensed. Did you understand? We call this as a chromosome. So chromosome ಆಗ್ಬೇಕಾದ್ರೆ ಎಲ್ಲಿಂದ ಫಾರ್ಮ್ ಆಯ್ತು? ಫಸ್ಟ್ ಡಬಲ್ ಸ್ಟ್ರಾಂಡೆಡ್ DNA, it will coil around histone protein to form nucleosome, and such nucleosome will form euchromatin, heterochromatin. Further they coil upon themselves to form, with the help of non-histone protein complexes, they form chromosome. So as they are compactly wound around protein, the 2.2 meter long DNA of one particular cell, each and every cell has 2.2 meter long DNA. It is compactly arranged in a micron sized nucleus. This is called DNA packaging. ಓಕೆ, ಇದರಲ್ಲಿ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಇದೆ. ಹಿಸ್ಟೋನ್ ತಗೋತಿರಲ್ಲ, ಹಿಸ್ಟೋನ್ ಇಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಬೇಸಿಕ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್, ಅರ್ಜಿನೈನ್ ಅಂಡ್ ಲೈಸೀನ್ ಅಂತ ಎರಡು ಬೇಸಿಕ್ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಇರುತ್ತೆ ಅದರಲ್ಲಿ. ಆಸ್ ಇಟ್ ಈಸ್ ಬೇಸಿಕ್ ಅಂಡ್ DNA is negatively charged and histone is positively charged, it can easily attach with the histone protein. Okay, please go through NCERT. If you have any doubts, in the next class we will catch up. Okay, thank you so much. Sir?
Okay, thank you ma'am.
Thank you sir. Sorry I was not able to complete the central.
ತೊಂದ್ರೆಲ್ಲ, ಯಾಕೆಂತಂದ್ರೆ ಮಕ್ಕಳಿಗೆ ಸೊ ಡಿಸ್ಕಂಟಿನ್ಯೂ ಆಗಬಾರ್ದು, ಸೊ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಕಂಟಿನ್ಯೂ ಆಗ್ಲಿ ಅನ್ನೋ ಒಂದು ದೃಷ್ಟಿಗೆ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಏನಾದ್ರು ಆದ್ರೆ ಆಪರ್ಚುನಿಟಿ ಇರ್ಲಿ ಅಂತ ಕೇಳಿದೆ.
ಖಂಡಿತ ಸರ್, ಖಂಡಿತ. ಎಸ್ ಸರ್.
Thank you ma'am.
Thank you sir. Thank you.
So I would like to thank you immensely for delivering a very informative and insightful talk on DNA structure. So on behalf of KST and myself, Thank you, ma'am.
Thank you so much, sir. Thank you.
ಸೊ ಇಂದಿನ ತರಗತಿ ಹಾಜರಾಗಿದ್ದ ನನ್ನ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು, ಅಧ್ಯಾಪಕರು, ಶಿಕ್ಷಕರು ಹಾಗೂ ನನ್ನ ಸಹೋದರಿಗಳಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಿದ್ದೇವೆ. ಹಾಗೂ ಎಂದಿನಂತೆ ಬರುವ ಸೋಮವಾರ ಡಾಕ್ಟರ್ ರೋಹಿತ್ ರವರು ಮತ್ತೊಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮ ನಡೆಸಿಕೊಡಲಿದ್ದು, ಎಂದಿನಂತೆ ಹೆಚ್ಚಿನ ಸಂಖ್ಯೆಯಲ್ಲಿ ಮಕ್ಕಳು ಹಾಜರಾಗಬೇಕೆಂದು ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ತಿಳಿಸುತ್ತಿದ್ದೇನೆ. ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್. ಥ್ಯಾಂಕ್ ಯು.
Thank you, sir.
ಸೊ ನೆಕ್ಸ್ಟ್ ಡೇಟ್ ನಾನು ನಿಮಗೆ ತಿಳಿಸ್ತೀನಿ.
Okay, sir. Okay, sir.
ಥ್ಯಾಂಕ್ ಯು.